Hello, everyone. Welcome back from wherever and whenever you are to another of our webinars from OpenEye Cadence Spykeller Sciences. My name is Paul Hawkins. I will be our compere MC for today. Today's presenter is Matt Geballe. He is a senior scientific solution architect here at OpenEye. He's been with us a long time. It's his 17th year at OpenEye. And in that time, he's filled a great many roles, including his latest one, which is to be the senior developer for the ROCS X solution, which is the topic of his talk today. Matt's title is Exploring the Uncharted, Discovery at Trillion Scale with ROCS X. Before I let Matt take over, a small housekeeping point. If you have any questions during the course of the webinar, please add them to the Q&A box on the right side of the screen. So with that, I will turn it over to Matt. Take it away, Matt.
Thank you, Paul, and for the introduction, and thank you everybody for joining us today. I'm talking about exploring the uncharted, discovery at trillion scale with ROCS X. So I think most of us can remember in our lifetime something that has gone from being hard to get to being readily available to being extremely abundant. And one example of such from my life that I was talking about with colleagues recently was with apples. So when I was a kid, if you had an apple for lunch, chances were it was a red delicious, which while red is not very delicious. And then over time, I started going to the grocery store and I started to see there were other kinds of apples available, things like Fuji's or Galas that were actually much more delicious. And of course, the holy grail was the Honeycrisp. But now when you go to the grocery store, there are too many varieties of apples, just a kind of an unending range of new names that I have no idea about. And actually there's websites you can find like applerankings.com that will rank over 90 different varieties of apples for you with some sort of cheeky reviews about whether they're good or not. Or maybe you could take an even more modern approach, and ask your friendly AI what kind of apple would be most delicious to eat, since AIs apparently know all about how apples taste. This kind of transition is something that we see over and over again, particularly in areas where technology is involved. So another one that we're probably all familiar is about video entertainment. So when television was first invented, right, there were 3 or 4 broadcast stations that you could watch, and they turned off in the middle of the night and the TVs just showed static. And then the age of cable TV came along and you had, 50 or 60 or even 100 stations that you could watch at any time. And you could look through the guide and decide, well, right now of all my choices, this is what I want to watch. Or you could, use the remote to channel surf, as many of us learned to do. But now we're in an area of there is too much video available to watch. There's streaming channels like Netflix and Disney that have tons and tons of shows for you to watch. There's YouTube where, every hour there are days worth of videos uploaded. And then there's, short form discovery based platforms like Instagram and TikTok that will just give you an unending stream of videos that you may or may not be interested in. If you're my daughters, you can just watch unending numbers of cute dog videos. And you can't watch all the cute dog videos on those platforms, but you can watch as many as you're interested in finding. We've gone through a transition like that in terms of virtual screening or hit identification as well. About 25 years ago, what people were screening were corporate collections, and they were maybe 1 million compounds, maybe 2 or 5 million at the larger end. And OpenEye provided a fantastic tool for doing ligand-based virtual screening in that arena, which was ROCS, where you take a ligand and we find similar ligands based on their 3-dimensional shape similarity and their 3-dimensional chemical feature similarity as represented by our color features. But about 10 years ago, we changed from wanting to search tens of millions to wanting to search hundreds of millions to billions of compounds. And this was really based on the rise of chemical vendors and sort of make to order compound libraries where you could search these large libraries, and vendors actually got very good at delivering these compounds with a high degree of success. And so in order to search hundreds of millions or billions, we took our ROCS technology, or our FastROCS technology, which is an implementation of ROCS on GPUs, and we moved it to the cloud. And so 10 years ago, I was going around giving talks about getting Orion into the 3 Comma Club, which was a joke based on the show Silicon Valley, which was on cable at the time. We leveraged the power of GPUs in the cloud, being able to get hundreds of GPUs if you wanted, and then shut them off when you were done, to be able to search billions of molecules using FastROCS and Orion. But that was 10 years ago. And in the meantime, purchasable chemical space has grown and grown. And the reasons why we might want to search beyond purchasable chemical space has also grown. So in the past, even 5 years, say, enamine real has grown from 1.4 billion molecules to 10 billion to 17 billion, and now around 25 billion compounds to search. And that's just one vendor. There are many other vendors that have come onto the scene that offer libraries of similar sizes. But part of this work that we did 8 or 10 years ago of being able to search billions really convinced us that there were also strong reasons to search outside of purchasable chemical space. So AstraZeneca published this paper based on being able to search billions of molecules with FastROCS on Orion that demonstrated that as you search larger and larger libraries, you get 2 really appealing effects. You find higher scoring molecules as you search larger and larger libraries, and the diversity of hits that you get above a given threshold increases as well. So those are strong reasons to want to search larger and larger chemical spaces, but also you might want to just be searching different areas of chemical space than all your competitors are. If you're all searching the same vendor libraries, then you can find all the same compounds. So one of the lessons we took from this is that part of the value of these vendor libraries was that these molecules are actually obtainable. They're not just virtual compounds that are in the computer, but there are compounds that you can order and get made, and they show up in a vial. And so as we thought about expanding the libraries that we search and building our own libraries, we wanted to keep this lesson about building libraries full of obtainable compounds close to heart. And so our approach has been to try to build the sort of next obtainable space beyond purchasable chemical space. And in our mind, this is what we would call easily synthetically accessible chemical space. And that really means compounds that you could make in one simple reaction from whatever things you have on hand. So purchasable building blocks or in-house building blocks. And so that is the strategy that we are going to take to address this area of being in, maybe having too many compounds to search. So vendor libraries are now into 20 billion or more, and we are building easily synthesizable libraries into the trillions from building blocks. So how are we going to search that? And that's really what ROCS X is about. It's about bringing the ROCS search that we are all familiar with and has been a proven gold standard for ligand-based virtual screening into this trillion compound arena. And this is the result of a multi-year scientific collaboration with Treeline Biosciences, where we learned how to build these libraries of easily synthetically accessible compounds in 3D because we want to search these larger libraries and see if we can see this beneficial effect of finding higher scoring compounds and more diverse hit lists. Our approach to doing this is to use multi-arm bandits and active learning. And that has resulted in this ROCS X product that I'm telling you about today and has just been released as part of our 2026.1 flow release. So now let's go inside ROCS X and talk a bit about how the search works. Because these libraries are built from building blocks, I'm going to use a sort of building block motif here in explaining that. But to understand how the search works, first you have to really understand how we build the library. So we're going to start there. So you have a set of building blocks of chemicals that you can use for synthesis. How are we going to build a 3D library that we can search? So the first step is we need to prepare these building blocks for 3D synthon search. And that means first recognizing, well, what sort of reagents could any molecule be used for? So if you had this molecule here shown in the middle, which you can think of as a simple building block, the first thing we need to recognize is, okay, this can be a reagent in a reductive amination, and particularly a ketone reagent. And then the next thing we want to do is to transform this from the chemical that you have, in stock to the way that it's going to be after it takes part in the reaction. And in this case, this means transforming it from what we would call a building block into a synthon. And that just means, in this case, removing the ketone and replacing it by an attachment group that we're going to use further on. But this introduces the first example of needing to deal with duplicates. And duplicates are something that can occur in many, many different steps in what we do in ROCS X. And we endeavor to treat them appropriately at each step for you. But the first place that duplicates can happen is in this building block to synthon transformation. So here's another molecule that we maybe can purchase from a vendor, and we would transform it into the synthon, we remove the halogen, and you end up with this synthon here at the bottom. But the vendor might supply a different molecule with an iodine instead of a bromine that can be transformed into the same synthon. And we don't want duplicate synthons in our search, but we want to understand that this synthon here can be arrived at from multiple different building blocks. And in fact, as we've built ROCS X to support multiple vendors and to track the provenance of that, different vendors might provide the same molecules, maybe with slightly different forms, like a salt form. And we want to only have one synthon in our library, but understand that where you defined a hit product that involved this synthon, it's really useful for you to understand, well, there's several different molecules that you could obtain from different vendors that would allow you to build this synthon into a product with a given reaction. Likewise, we also need to think about deprotection, and that, in fact, leads to more synthons and more opportunities for duplicates. So sometimes building blocks have protecting groups, such as in this example here, and when we process this into a synthon, we remove the boronic acid, but the protecting group remains. But given that we're trying to transform the building block into the way that it would appear in a product molecule, one of the things that you could do is you could do the synthesis and then remove the protecting group. So in this case, we recognize the protecting group and process this building block into two different synthons, one with the protecting group present and one with the protecting group removed. So overall, the approach for building a library for ROCS X is a three-step process. But we provide a flow that runs these multiple stages for you so that it's a very simple process if you want to prepare your own library, which is something that you can do with the ROCS X flows. So the inputs that you require are a set of building blocks, either from a vendor or from your in-stock supplies. And the first step is we pass these through our reagent definitions and this reaction infrastructure that we've built up over several years and use in other generative design flows in Orion. In ROCS X, we support about 35 two-component reactions, and we pass all these building blocks through them and classify them into the different sorts of reagents and build that into a reagent database. In the second step, we transform the reagents from that into synthons and do deprotection and build those into a 2D synthon library. And in the third step, we do deduplication that we talked about, and then we do conformer generation using our conformer generator Omega so that we can do 3D searching. We also sample some products from combining these reagents and generate conformers for those products too that we want to use in the different stages of the search that I'll talk about in a few minutes. So One of the challenges about preparing libraries is the cost. When we generate conformers for the enumine real database, which is 24 billion molecules, which this is something that we do and make these available for our customers, but it's a significant amount of calculation. It costs about $50,000 to do that preparation, and it takes a significant amount of time, partly because we break it up into chunks and do it in pieces. One of the advantages of moving to combinatorial libraries is that the preparation of the libraries is much cheaper and much faster. So in ROCS X, we ship with 2 different libraries that are full of easily synthetically accessible compounds. The first is built from a set of purchasable building blocks from 3 different vendors, Enamine, MQL, and Molport. That was about 7 million SMILES at input. It builds a product space of about 3 trillion product molecules, and the cost to build that is only about $150 if you were to build that yourself. We build a larger library, also filled of easily synthesizable compounds from purchasable molecules, and we got that by combining the building blocks and the in-stock screening libraries from those 3 vendors as well. That started as about 26 million SMILES, builds a product space of about 11 trillion molecules, and costs a little over $200 to prepare. Those are the main libraries that we ship with ROCS X. You'll see those again in the validation work that I talk about later in the presentation. But we also wanted to sort of push the bounds a bit and see, well, if we wanted to build larger and larger libraries, is that possible? And so we've played with subsetting the enumine fast-made set of building blocks, which starts as 93 million SMILES. And we subset that in some different ways, and we're able to build libraries containing 65 trillion products and over 200 trillion products for a very reasonable cost and time, with some sort of extra experimentation and by skipping the product sampling that I talked about. These are very functional libraries. We can search these, and I think it's a good demonstration that we can build even larger libraries than we've built to date and search them. But searching these libraries now is the question. We can build much larger libraries. How are we going to search them and find the right things out of these enormous chemical spaces? And so now let's talk about the ROCS X search process. And to use this, I'm going to stick with the block analogy. I've invented this weirdly shaped block query here that, I don't know what it looks like. Maybe some sort of duck snake wearing a party hat, perhaps. And let's say, how are we going to find things that can create this shape? And one of the things that we sort of stuck with as we built ROCS X is expressed by this quote from Charlie Munger, who is the vice chairman of Berkshire Hathaway until he passed away in 2023. And he says, I try and keep it simple and fundamental as much as I can. And we know a lot fundamentally about finding similar molecules. ROCS and FastROCS are fantastic tools that we've used for decades for this purpose. And so the way we built ROCS X is to really leverage ROCS and FastROCS in many, many different ways to build this combinatorial search. So let me explain how that works. So if we wanted to find our strange query here shown in the top right, the first thing that we do is a simple FastROCS search to say, well, what other similar molecules can we find that are sort of like this query? And that FastROCS search can be done on the sampled products from our library that we built into our search library, or it can be from a search of any other enumerated library. Then we take this set of molecules that are similar to our query, and we fragment them and find the unique shapes among those fragments. So we do those comparisons with ROCS to find the unique shapes, and we end up with a set of unique shapes, and we call these shape arms because they are going to be the arms in our multi-armed Bayesian bandit system. But the next thing that we want to do is we want to find, well, what synthons do we have that are similar to these shapes? So we take these shapes and we search them against our synthon library, and for each shape, we come up with a list of synthons that are similar. And this has the effect of focusing our Synthon library, limiting the space within our Synthon library that we're going to do further searching. You can do this by selecting which vendors you want to search. So ROCS X allows you to limit your search by vendor at search time. But we would estimate that this reduces us to looking at maybe 5 to 15% of the total product space. And this is really part of the power of searching this large space is, if we go back to another quote from Charlie Munger, this is about fishing where the fish are. This is about limiting the enormous trillion molecule product space to a smaller product space that we are going to then fish in. So the next step of this is to then try combining these different synthons and then compare those combinations to our query. And this is done using Omega to generate conformers and then ROCS to compare to our query. But we want to learn as we go about which combinations work and which don't. And so this brings in our multi-arm bandit with our active learning component, which we kind of think of as an octopus playing several slot machines at once. So we sample many different combinations of products. We see which ones build good combinations in terms of comparison to the query or not. And we learn as we go so that future sampling takes advantage of what we've learned. And so again, as Charlie says, we are just trying to learn all the time as we sample these products. So to walk through this in a maybe more traditional schematic, not using building blocks, this is what the ROCS X search looks like. It's also a 3-step process. We also provide a multi-stage flow that runs all 3 steps for you. So all you need to do to execute a search is to bring your query and to select which 3D library you want to search. In the first stage, we do that initial search using FastROCS, get that initial hit list of similar molecules, and then we work on initializing our Bayesian bandit model by discovering our shape arms and searching them in Synthon space. Again, that uses FastROCS. We estimate we do hundreds of billions of comparisons at least in initializing the model and focusing our search. In the second stage, we do sampling out of our Bayesian bandit model. We sample products, we generate conformers and evaluate them using traditional ROCS, and then we feed back that evaluation into our active learning cycle to improve future sampling. At the end of that, you get a ROCS X hit list, which contains regular ROCS results. And then in the third step, we provide some hit list triage. We cluster that hit list using fingerprint clustering and give you a fully clustered hit list as well as a sampled hit list aimed at giving diverse results. So once again, we have to worry about duplicates because even though we've done all the deduplication work in building the library to make sure we have unique synthons, you can still end up with duplicates in your products. Here's an example of a product molecule that we found in one of our searches, and it was formed by combining these 2 synthons that you see here. There are actually 2 different reactions where we could combine these synthons, and some of those synthons are available from multiple vendors. But we also found that product molecule by combining 2 completely different synthons. And so while we do the deduplication to make sure this product molecule only shows up in your hit list once, it's really valuable information were you to be interested in actually making this compound, that there were multiple synthetic routes from different kinds of starting positions or different starting structures that could get you to this product. And so all of that information is available to you in the ROCS hit list, a ROCS X hit list. Okay, so now let's shift from how does the search work to how well does it work. Let's analyze the results and the performance of these of this new ROCS X search. So to do that, we're going to look at 15 validation queries. And these were chosen to be a shape diverse set across 5 different targets where we picked 3 queries from each target. These are all queries from crystallographic conformations of these ligands. And this is out of a data set that was curated for our 3D QSAR work that you may have heard about in other recent presentations. So the way that we looked at validating ROCS X is to compare the ROCS X search first to a FastROCS plus search of enumerating real database. So we are not searching the same library in this case. So this raises some important questions about how we think about validation, because it's not an apples to apples comparison of searching the same library and then seeing if you found the same things. So we're looking at things like, how does the range of scores within a hit list compare? Also, because ROCS X is probabilistic and we do a fixed amount of sampling, we'd like to understand how does the amount of sampling that we do affect the hit list. And then, there's the kind of the fundamental question of, when you do a probabilistic search, and especially at such large library sizes, do you find the best things that you could find? And how can we actually know that when, trillion molecule libraries are too large for us to do an exhaustive comparison? So in terms of ROCS X searches, initially we do some comparisons to a 1.3 trillion molecule library that we've prepared. These comparisons are all sampling 1.3 million products. So we're only actually sampling and evaluating 0.0001% of the molecules in that product space. And in this case, we are building hit lists of about a little over 32,000 molecules, which is an even smaller, nice round percentage of 1.3 trillion molecules. Later, when we built the 3 trillion and 11 trillion molecule libraries, we'll do some comparisons of those. In those cases, we're doing 1.5 million samples in most cases. And there we are looking at truncating hit lists based on a score threshold that we've determined from the bottom of the 10,000 molecule FastROCS hit list. So when we look at these hit list comparisons, we're going to be looking at lots of plots like this. This is what we call a rank score plot. So in this case, we're looking at one of these initial hit list comparisons where we're comparing hit lists of a little over 32,000 molecules each. We're going from the top of the hit list on the left, or rank 1, all the way down to the bottom of the hit list on the right, or rank 32,500 on the x-axis. On the y-axis, we're showing the tandem-modal combos of those hits between these two different searches. So in the teal, we're looking at the FastROCS plus search of enumerating real database, so 24 billion molecules. And on the right, we're looking at our search using ROCS X of the 1.3 trillion molecule library, again, doing only 1.3 million samples there. And so what we can see, this is a search of one of our queries, a relatively small query. And we didn't know this when we chose this query, but this query molecule is actually present in both the enumerating real database and in our 1.3 trillion molecule product space. And both FastROCS and ROCS X find the query molecule in the library and put it right at the top of the hit list with a tandem-modal combo close to 2.0. So you can see the top ROCS X result here shown in gray compared to the query shown in green. It looks like a regular ROCS result because it is a regular ROCS result. Once we sample this molecule out of the library, we generate conformers for it and evaluate it with ROCS. But we do see that over the course of going through and down the hit list, the molecules we're finding with the ROCS X search are higher scoring in terms of tandem-modal combo than the molecules we got from the FastROCS search of enumerating real. And given that this is a regular ROCS result with a regular tandem-modal combo, we can apply, all the body of work that's been done over the many years that we've been working with ROCS and tandem-modal combo to understand these results. And one of the really powerful things there is this work on belief theory that was done by Steve Muchmore and others at Abbott in the 2000s. And what they showed is that you can actually take the tandem-modal combo between a query and the hit molecule you found and correlate it to the probability that your hit molecule has the same biological activity as your query molecule. So if we were to just look at a simple cutoff and say that around tandem-modal combo of 1.7, that corresponds to about a 40% probability that the molecule you find has the same biological activity as the query, we can sort of quantify that the benefit of this higher scoring hit list is with the ROCS X hit list, you've got about 15,000 molecules that have a 40% or better probability of being as active as your query. Whereas in the FastROCS hit list of enumeration real, you've got about 2,000 or 3,000 molecules that have a 40% probability or higher of similar activity to your query. This is a very small and simple query. We move on to something a bit more complicated, like this molecule here. We see again between the searches of enumeration real with FastROCS and the 1.3 trillion with ROCS X that now we've got separation from the top of the hit list all the way down to the bottom, where ROCS X is finding higher scoring molecules. Here's the hit that's actually at the top of the ROCS X hit list, so you can kind of eyeball the reasons why this would be similar in terms of shape and color. Here's the top thing out of enumeration real. Still has some similarity, but a bit lower similarity as represented by the tandem-modal combo. Here's the overlay of the ROCS X hit to the query shown in green. So you can see the 3D shape and color similarity represented there. And if we take the belief theory curve again and apply it to these two hit lists, but not with a sort of simple cutoff analysis, but by doing something a bit more complex, where we're going to take each molecule in the hit list and basically do a Bernoulli trial for each molecule. So think of it as doing a coin flip, where the probability of your coin being heads is the probability of activity that comes out of this belief theory curve. We can take these two hit lists, and by doing this sampling, we can estimate that this increase in tandem-modal combo in the ROCS X hit list actually gives you about a 3 times increase in the expected prevalence of actives via belief theory. So finding these higher scoring hit lists has a real improvement in your chances of finding an active compound, at least as active as your query via belief theory. So what about a scenario where ROCS X doesn't do as well? So here's a query that's a bit larger and a bit more complex. And when we look at the curves from the FastROCS hit list and the ROCS X hit list, they're pretty similar. ROCS X is a little bit higher at the beginning, but they sort of cross over a bit. And nothing that they're finding is that similar. The top hit here, tandem-modal combo, a little above 1.25. You see the top ROCS X hit shown here in 2D and then shown in gray with the 3D overlay. And you can see it's matching the shape and chemical features quite well in some portions of the molecule, but other portions of the query, it doesn't match at all, or the match there is not so good. So in the exhaustive search with FastROCS, we know, okay, there's just nothing in enumeration real that is that similar to this query. Now, ROCS X is a probabilistic search, as we talked about. So while we've seen evidence with other queries that it does quite well at finding the best thing, we don't know for sure that there aren't other molecules in this library that are more similar that we just didn't find. So one of the things that you could do is you can just sample more molecules with ROCS X. And so when we do that here, this is doing 6.5 million samples rather than 1.3 million samples. So 5 times the amount of sampling. And what we see is that while there's a small improvement in the hit list shown now in gold in the rank score plot, there's not really a sort of great large impact. So doing much more sampling in this case, we didn't really find anything that was dramatically better than what we had found before. And we can't do the exhaustive search of 1.3 trillion to know for sure, what are the best things in there, and did we find them? But what we can do is we can add in some things that we know are good and then see if we're able to find them with ROCS X. And so that's what we did by taking the query and some other actives for this target and fragmenting them into synthons using retrosynthetic transforms from our reaction definitions. So we broke these molecules up into synthons, and then we sprinkled in these, needle parts, if you will, into our haystack, and then redid the ROCS search to see, well, if we know there are some good high-scoring molecules in there to find, do we find them? And this is the hit list shown in gray now, of ROCS X on the 1.3 trillion molecule library with these active synthons sprinkled in. So we see much higher scores across the board. We see that ROCS X was able to find and prioritize building synthons from these active synthons, the active molecule synthons. And up at the top of the hit list, we see with a Tanimoto combo now approaching 1.8, so not 2.0, but very close. And those differences are really because of the differences in conformation between a crystallographic conformation and an omega conformation. At the top of the hit list is the query molecule that we're able to form from the synthons that we placed into the library, and ROCS X was able to find that and pull that out and have it at the top of the hit list, as you would expect. So even though we're doing a probabilistic search, we have some strong evidence that we are able to find many of the best things there are to find in a library, especially when we insert some things that we know are good. If we want to think about generally how reproducible these results are over multiple trials, we've done some experiments along those lines as well. Now we're looking at repeated searches of the 3 trillion molecule library. And what we see here is if we do 3 different searches at 500,000 samples with the small dashed line, there's quite a bit of variability in the rank score curve. And that goes down as we go to 1.5 million samples and then to 5 million samples. This is very query dependent. So some queries show more variability, other queries show less variability, as you see here. But overall, we can say about 60% of the hits are in common between any 2 repeated searches at the 1.5 million sample level. So there could be reasons to do multiple searches if you're worried about reproducibility, but it's quite high. We also looked at the effect of searching different and larger libraries, and now we're switching to the different score regime that I talked about before. So here we took a query and did a FastROCS search on enumeration, which is shown here in teal. And then we took the bottom Tanimoto combo from that 10,000 molecule hit list. So in this case, around 1.1 Tanimoto combo. And we used that cutoff to see, well, how many molecules do we find when we search a larger library with ROCS X above that threshold? So for this query, when we searched the 3 trillion molecule library, we found about 18,000 molecules above that Tanimoto cutoff. So getting close to twice as many molecules from searching this larger library. When we went to searching the 11 trillion molecule library, here we found many, many molecules above that threshold. In fact, over 100,000. We cut off the ROCS X hit list at 100,000. And even at the bottom of that 100,000 hit list, we were still finding things that were above the Tanimoto cutoff found from the 10,000 molecule hit list. So if we take this approach and do it across our 15 queries, we can generate a plot like this, where we're comparing the number of hits found above that cutoff for FastROCS on enamine real, and then ROCS X on the 3 trillion and ROCS X on the 11 trillion. Remember, we derive the cutoff from the FastROCS library, which is why those are all fixed at 10,000. And so we can see across these queries, searching larger libraries are giving us more and more higher scoring molecules. We can also look at the maximum Tanimoto combo. So what is the score of the best molecule that you found? And we also see, as we go from searching enamine real to searching larger and larger ROCS X libraries, we do see an increasing top of that hit list. So we are finding higher and higher scoring molecules from these searches. So focus on this plot on the right, and we shift it over here. You might recall from the beginning of the presentation that this echoes one of the important aspects we saw from the work that AZ did and published 8 years ago, that as you search larger and larger libraries, you find higher and higher scoring molecules. We are seeing that effect with ROCS X as we search larger and larger libraries. Even though we're no longer doing an exhaustive search, we're doing a probabilistic search. We're still able to draw more and more higher scoring molecules out from these larger libraries. So we might move from doing this sort of straight up comparison of looking at the distribution of these queries to doing a pairwise analysis. So now we're looking at, for each query, how does the number of, or the highest Tanimoto combo change as we go from library to library. So FastROCS has disappeared from this results, and now we're looking at deltas in the mean or the maximum Tanimoto combo as we search larger and libraries. And so this just recapitulates what is clear from the graph on the left, that searching larger libraries is giving you more and higher and higher scoring molecules. And we take this pairwise analysis, and if we also apply it to the second learning from the AZ work, that searching larger libraries gives you more diverse hit lists, we can see that replicated here as well. And depending on what your metric of diversity is, whether it's number of Bemis-Merkle scaffolds or number of hetero Bemis-Merkle scaffolds, or just the number of clusters you get from doing a fingerprint clustering of the hit list, we see that above that threshold, searching larger and larger libraries is on average giving us more diverse hit lists, independent of your measure of diversity. So searching these larger libraries is great, but what about the cost? FastROCS, which we use to search enumerating real, is very, very cost efficient. We're able to search 24 billion molecules exhaustively for a cost of about $340, $350. The time can vary a bit, mostly based on GPU availability. We all know these days with the rise of AI, it can be difficult to get your hands on GPU sometimes. But one of the real advantages of working with combinatorial libraries and ROCS X is the cost effectiveness of the search. So we can search libraries, 2 orders of magnitude larger than 24 billion for about an order of magnitude less in cost. Searches of the 3 trillion molecule library take $25 to $30. Searches of the 11 trillion molecule library take $40 to $45. And this even scales well into the very large experimental libraries that we built, where we would estimate that searches of 65 or even over 200 trillion molecules could be done for hundreds of dollars. So we're excited about the possibility of pushing this into larger and larger, even theoretical virtual spaces. So to conclude, ROCS X brings a ROCS search that you may be familiar with, that you trust, to trillion scale molecule libraries. The ROCS X hit lists that we get are statistically significantly higher scoring than the FastROCS searches of enumerated libraries. When we seed in actives, ROCS X is able to recover them, and the ROCS X hit lists show the increased diversity that we expect, given that we're searching larger and larger libraries. Generating these libraries and searching them are very efficient compared to working with enumerated libraries. And we've built these libraries to be full of synthetically accessible compounds, and our partners report a high degree of being able to actually source these compounds. And they have sourced and had made hundreds of molecules that weren't in vendor libraries that came from these combinations of building blocks. And it's available now with the 2026.1 flow release. So in conclusion, ROCS X lets you search much larger libraries, helps you find better hits. You get the same ROCS scores that you're familiar with, and the cost is way less than the enumerated searches. With that, I'd like to thank people who were instrumental in building this. First of all is Alex Lee, who did most of the development of the multi-arm Bandit and active learning cycle. Bert Leland was responsible for the library generation work. A lot of thanks to the larger science and cloud teams at OpenEye, particularly Jingyi Chen, who curated the data set that we ended up using for our validation ligands. Thanks, of course, to our leadership here for funding and encouraging this project. And thanks to our scientific collaborators at Treeline, particularly Eric Manis, Brian Cole, and Andrew Fenley, who provided a lot of input and guidance over the several years that we worked on this together. with that, thank you. I hope you enjoyed this webinar, and I'm happy to answer questions now, or please send your questions in later.
Thank you as always, Matt. Magisterial, as we've come to expect. A lot of questions, as you may expect, interesting to a lot of people. So we're probably not going to be able to go through all of them. So just to reiterate what you said, if you in the audience have any further questions, please do email us and we will reply to those as soon as we can. So to start at the top of the list, first one in, you mentioned at the beginning that you're using 2 component reactions to enumerate the libraries. I'll be able to use 3 component reactions or more.
So not currently. Right now, we just support 2 component reactions. It is pretty high on the list of things we'd like to add now that we've got the 1.0 out. We were unsure of how large libraries we could get with 2 component reactions. Turns out you can build very large libraries just with 2 components. But once we get to 3 components, that will kind of just juice the combinatorics even more. So it's something we're definitely excited to get to.
Very good. This feels like a question from someone with a real interest in the actual chemistry, the actual synthesis. Do you support deprotection reactions in the library building?
So we do support deprotections. They are supplied as a set of transforms that we apply to the synthons, as I mentioned in the slides. We have an example set of deprotections that we've made available and that we've used to build the libraries that we supply, but they're fairly straightforward to add or edit that set of deprotections yourself, especially if you're going to build libraries using in-house building blocks. There's so many different variations of protecting groups. We definitely would encourage people to, customize that file and make sure they are getting all the deprotections that they want to have.
Very good. Watch this space. Okay. One on the more technical side, how accurate are the predictions that ROCS X produces of ROCS scores from the sample?
It's a good question, and I think it's important to understand because I feel like it's one of the powerful aspects of this approach is we aren't actually predicting the ROCS scores at any stage in the process. We use ROCS to build our model, and we use ROCS to determine which synthons are likely to provide good combinations. But when we evaluate a product molecule, we are doing exactly what would have happened if that molecule were in an enumerated library. We take the smiles and we generate conformers with omega, and we compare those conformers to the query using ROCS. And so that leads to, I think, one of the powerful things, which is the results you get are equivalent to the results you would have gotten had you built an enumerated library and evaluated that molecule. And then we can, rely on the tandem combo, a metric we know and trust, and have a deep understanding of the implications of.
So same answers, but faster.
Right. Who doesn't like that?
Everybody likes that. So here's another, again, on the library chemistry side. You spoke about initializing the search with searches from a collection of compounds. Do you need to search products from the library that you're wanting to search as part of that initialization process? Or can you use other sets of molecules instead?
Yeah, so no, you don't need to search products from that library as part of the initialization. We've built that into the library generation process, basically just to make a better user experience for doing the search. If you have a query and you pick one of our libraries to search, we have everything we need to execute that search for you. And that includes doing that initial search to build that initial hit list. But we do recognize if this is a query you're interested in, chances are you've probably done some regular FastROCS search, maybe used molecule search in the Orion UI to do a very fast search. So you might have a hit list of already known similar molecules, and you can provide that hit list, and we can either merge that hit list with the hit list of products and take the best things, or you can just skip the product search and use the hit list you've provided to initialize your model. So I think there's a good opportunity for you to bring things you've learned from other searches. And I think that means there's good reasons to do exhaustive searches of enumerated libraries and do ROCS X searches of these larger chemical spaces as well.
Well, Matt, thank you. I think that brings us to the end of the time we have, so I will bring the Q&A to a close here. And again, we will respond to any questions we didn't get to during the live part of the Q&A by email as soon as we can. So it just remains for me to thank Matt very much for the presentation. Thanks, Matt.
Thank you, everybody. And look forward to talking to you all again soon sometime.
So thank you, everyone, for attending the webinar today on ROCS X. I hope you found it as useful and as interesting as I did. We will, of course, be producing more webinars in the near future, so please do keep an eye out for notifications for the next email, and we look forward to seeing all of you then. In the meantime, thank you and goodbye.